Welcome to this taster lecture for MSc Medical Microbiology. My name is Dr. Simon Baines and I'm a Principal Lecturer in Microbiology at the University of Hertfordshire. The lecture today is going to consist of around 10 minutes, and what we're hoping by the end of the lecture is that students will be able to discuss the scale of antimicrobial resistance around the world and also discuss one particular resistance mechanism around vancomycin resistant Enterococci and their resistance to glycopeptide antimicrobials using the VanA system. The world faces a huge global challenge in the form of antimicrobial resistance. This is not a new challenge. We can see from the screen there are World Health Organization data and quotations around the scale of antimicrobial resistance that are over a decade old. However, we still face this huge challenge because we have not made huge progress in the scientific community in tackling this challenge of antimicrobial resistance in the past decade. You can see from the right hand side of the slide some of the numbers associated with antimicrobial resistance. 25,000 people per year die in Europe as a consequence of antimicrobial resistant infections. There are a similar number in the United States of America as well. Contributing factors to antimicrobial resistance include the inappropriate use of antimicrobials in general practice. You can see around 40% of prescriptions given in general practice for antimicrobials are inappropriate. There is huge antimicrobial use in the agricultural sector, as well as antimicrobial compounds being used in the home environment and veterinary environments as well. It's been a very long time since we've had a brand new novel class of antimicrobials produced. And this again compounds the problem of antimicrobial resistance and means that we have to rely on existing antimicrobials. There's a huge financial burden for the problem of antimicrobial resistance. You can see 1.5 billion euros per year of additional healthcare spend due to antimicrobial resistant infections. Global deaths now are around 700,000 deaths per year, and if we do nothing about the problem of antimicrobial resistance, then it's estimated by 2050 that there will be an additional 10 million deaths per year, and the cost to healthcare systems around the world will be 63 trillion pounds. So you can see the scale of the problem is huge, and we have to tackle it using a multifactorial approach. The beeper that you heard at the start of this session was designed to give you an idea of how many deaths may actually result from antimicrobial resistance by 2050 if we don't do anything. Those beeps were every 3 seconds. So every 3 seconds by 2050, if we don't take any action, one person in the world will die due to an antimicrobial resistance infection. In terms of the significance of antimicrobial resistance by 2050, if we take no action, there will be more deaths due to antimicrobial resistance than there will be to cancer. So if you can go to mentimeter.com and insert the code you can see on the screen, there's a survey up there that I'd like you to complete, just to give me an idea of your background knowledge on the subject of antimicrobials and antimicrobial resistance. So first question, antibiotics are drugs that can kill or inhibit microorganisms and include naturally occurring compounds, synthetically produced compounds, semi-synthetic compounds, or all of the above. So think which answer you feel is correct and insert the answer into the Mentimeter program. So I can see from your responses that 82% of you voted for all of the above. Now, actually, antimicrobial compounds, such as antibiotics, vary in their definition. So antibiotics are actually the naturally occurring compounds only. Okay, we see antibiotics referred to in the press and in the scientific literature regularly, but they're often misquoted. So if you are going to discuss and write about antibiotics, then you are dealing with only naturally occurring compounds. If you would like to discuss synthetic or semi-synthetic compounds, or in fact any antimicrobial compounds, then you would use the term antimicrobial substance. So how many years old do you think the oldest antibiotic-resistant bacteria that have been discovered by scientists are? Take a moment and insert the answer to that question into Mentimeter. Okay, your responses are in, and I can see that we've got a variety of responses, some of which are shown on the screen now. You'll actually be interested to know that the oldest antimicrobial resistant bacteria discovered are those which have been discovered in permafrost. That was 30,000 years old. So bacteria have been recovered, antimicrobial resistant bacteria from permafrost, and they're 30,000 years old. And this vastly predates the antibiotic era. So this should tell you that the problem of antimicrobial resistance is not a new one. It's a naturally occurring phenomenon whereby bacteria in the environment develop mechanisms to become resistant to antimicrobial substances in order for them to survive exposure to those antimicrobial substances and go on to reproduce. So if we look at genetic data and if we consider phylogenetic analysis of beta-lactamase enzymes, we can estimate just how old antimicrobial resistance mechanisms may be. So there's a question on the screen for you. Phylogenetic analysis of beta-lactamase enzymes responsible for resistance suggests that they are approximately how old? So if you head to Mentimeter and insert the answer that you feel is most appropriate, either 1,000 years, 10,000 years, 100,000 years, 1 million years, or 1 billion years. So your answers are in. I can see there's a good spread of answers. However, beta-lactamase enzymes are estimated to be 1 billion years old. In fact, 1.5 billion years old. So you can see that these kinds of enzymes have been produced to deliver antimicrobial resistance and to allow survival of microorganisms for an incredibly long period of time. So as scientists, we are battling natural processes, natural evolution of microorganisms, which is a very difficult process to battle. We have compounded the problem of antimicrobial resistance by providing that selection pressure for antimicrobials, by using them clinically, by using them in the veterinary world, and by using them in the agricultural setting. And our elevated use of antimicrobial substances has actually forced the problem of antimicrobial resistance into clinically significant pathogens, which therefore compounds our ability to treat infections with those pathogens. So if we're going to consider antimicrobial resistance, it's very helpful to have a good definition of what actually antimicrobial resistance is. And Alistair McGowan and Wise produced what I think is the best definition, and it is the reduction in susceptibility from a wild type distribution of susceptibility for a species in a defined phenotypic test system. So whenever a laboratory defines if an organism is resistant or not to an antimicrobial substance, it is done in a defined phenotypic test system. And what this ensures is that we use testing systems in different laboratories which are reproducible and very controlled in the conditions so that we get a result which can be compared between different institutions when they're doing their susceptibility testing. Now, as you'll know, Resistance can be intrinsic in that an organism does not possess the target for an antimicrobial substance, or the organism may be impermeable to that particular drug, and this is a natural inherited system whereby the progeny of a microorganism all share that ability to be intrinsically resistant. So this could be gram-negative bacteria being intrinsically resistant to penicillin G, or it could be that bacteria are not susceptible to antifungal agents because they don't possess the target for them. Resistance can also be acquired, as you know, and this results in the emergence of resistant bacteria, and it's a consequence of the change in the physiology or structure of that microorganism caused by genetic changes. Now, this could be in an individual when they are receiving antimicrobial therapy. It could be in the environmental setting whereby an organism is exposed to an antimicrobial substance and there is a selection pressure for the development of resistance, and one member of the population may mutate as a consequence of natural mutational events, and the selection pressure in the environment ensures that that resistant mutant is selected for. The susceptible population of the bacterium may die, and there may be expansion of that resistant mutant. Now, if we consider glycopeptide antimicrobials, these are a very important class of antimicrobial substances. And they interfere with the replication of bacteria by interfering with cell wall synthesis. And they inhibit both transpeptidation and transglycosylation reactions by binding to the terminal elements within peptidoglycan. So you'll know that peptidoglycan is a substance unique to bacterial cell envelopes, and inhibiting that process allows us to deliver selective toxicity against those microorganisms. They're very large antimicrobials. You can see 1.5 to 2 kilodaltons in size, and their spectrum of activity is mainly gram-positive bacteria. They're too big to get through the cell envelope of a gram-negative bacterium. The best example in this class is vancomycin. This is a very extensively used antimicrobial. It's been around for over 60 years. However, it's still a very important drug for us. In terms of how it works, Patrice Courvalin in 2006 reviewed the mechanisms and resistance to glycopeptide antimicrobials. So if you feel like doing some further reading, that's a very good review article to read. Although it's nearly 15 years old, it's still very pertinent. In the cytoplasm of bacteria, the organism begins to produce the precursors for new peptidoglycan, which must be produced in the cytoplasm and moved across into the actual growing cell wall. And you can see that there are various enzymes involved. We have race-mace enzymes, which are involved in making D-alanine, and we have ligase enzymes, which are involved in bonding these residues together in order that they can be attached to the growing peptidoglycan chain. The units which are involved in peptidoglycan structure are N-acetylglucosamine and N-acetylmuramic acid. There are alternating residues of these two carbohydrate structures, and at the end of those structures we have the terminal D-alanine D-alanine residues that would be produced during normal peptidoglycan synthesis. The lipid carrier C55 moves these peptidoglycan precursors across the bacterial cell membrane and transfers them into the growing peptidoglycan chain. Now, what glycopeptide antimicrobials do is they have a large cleft in their structure which binds to the terminal D-alanine D-alanine residues, and that binding prevents the incorporation of the peptidoglycan precursors into the growing peptidoglycan structures within the cell wall. So it inhibits transpeptidase reactions and transglycosylase reactions. So how does resistance occur to glycopeptide antimicrobials? Well, organisms can produce altered peptidoglycan precursors such that glycopeptide antimicrobials do not bind to those structures with sufficient avidity in order to inhibit peptidoglycan synthesis. So this could be, rather than having a D-alanine D-alanine terminal peptides, we might have D-alanine D-lactate terminal structures, and that may confer resistance. It may be that an organism is able to overproduce peptidoglycan, as has been seen in some Staphylococcus species. There have not been any reports of the breakdown of glycopeptide antimicrobials. So you may have read some examples of organisms which are able to overproduce peptidoglycan, such as Vancomycin intermediate Staphylococcus aureus, VRSA, or Glycopeptide intermediate Staphylococcus aureus, also known as GISA. And the overproduction of peptidoglycan means there is more of that target for binding, and therefore glycopeptides can't bind sufficient quantities of that peptidoglycan precursor, and therefore cell wall synthesis can still remain. If we look at that in a little bit more detail, you can see some electron micrograph images of Staphylococcus aureus, which have varying levels of resistance. We can see in A we have a wild type. Now the wild type organism has a normal thickness to its cell wall and is susceptible to glycopeptide antimicrobials. At the top of the screen here we can see what a resistant organism may be defined as. So a minimum inhibitory concentration of 4 to 8 milligrams per litre would be classed as a vancomycin intermediate susceptibility organism, whereas a resistant organism would be classed as greater than or equal to 16 milligrams per litre for its MIC to vancomycin. You can see that organism B in this image here is a vancomycin intermediate strain. You can see how much thicker the cell wall of this organism is. Heteroresistant organisms are where we have some members of the microbial population which are resistant. These can have thicker cell walls as well. You can see organism C here has got a thicker cell wall structure, and it's possible that those heteroresistant organisms under an appropriate selection pressure can expand clonally and therefore deliver resistance in that way. The VanA system, which originated in Enterococcus species, has now been identified in Staphylococcus aureus, in MRSA. This is a plasmid-mediated system, which is potentially a large problem for treating gram-positive infections. It's been identified in various countries around the world. It's been in Enterococci for some time. However, the movement of this resistance element into Staphylococcus aureus is a large concern for clinical treatment of infections. If we have a little bit of a closer look at the types of molecular systems which are responsible for vancomycin resistance, the table on the slide shows glycopeptide resistance mechanisms which have been identified in Enterococcus species. So if we look just at the VanA system here, it's been seen in Enterococcus faecium and Enterococcus faecalis. It can be plasmid mediated or chromosomally located. However, it has moved on to plasmids and has moved into Staph aureus, as we just said. It's inducible in that the system requires the presence of a glycopeptide in order to deliver resistance, and it delivers resistance to a range of antimicrobials, including vancomycin and ticaplanin, and its high level resistance. You can see that there are varying other phenotypes of vancomycin resistance elements in Enterococcus species, but we're not going to go into those in this session. So if we look a little bit closer at the genetic elements involved, we can see there are a large number of genes involved in this VanA resistance phenotype. There's a two-component sensor kinase system. So if you remember from the previous slide, we said that it's an inducible system. Therefore, presence of the glycopeptide in the environment is sensed and responded to by the two-component sensor kinase system. What it actually does is the VanA element itself is a ligase which is able to incorporate D-lactate into the terminal elements of the peptidoglycan precursors to produce D-alanine D-lactate rather than D-alanine D-alanine. Therefore, peptidoglycan synthesis can continue even in the presence of vancomycin or other glycopeptide antimicrobials. Okay, so that's the end of this taster lecture. So we've learned a little bit about the scale of antimicrobial resistance in the world, what a huge problem potentially it could be if we take no action. And we've learned a little bit of detail around glycopeptides, how they work, and the types of resistance mechanisms that organisms can have. What I'd like you to do by the next workshop that we have is to review the article that you can see on the screen here, which is looking at Staphylococcus aureus and vancomycin resistance, and we will discuss this in the workshop that will follow this lecture. So thank you very much for your attention.